Very good evening to all the NEET aspirants. This is your Prasanna Ma'am from NEET World. So yes, in today's class, we are going to learn about nucleic acids in detail, connecting the dots with molecular basis of inheritance also. So yes, let's get started. Before going into the concept, let me introduce you the NEET World special programs. For 11th standard students, you land on NEET World's 2-step to medicine. Whereas for 12th standard students, you will land on the program NEET World's 1-step to medicine, and for the students who are going to take long term, you will step into NEET World's power step to medicine. Whatever the program it might be, you have the same fee structure, that is just 3333 per month, where you can get access to with the complete lecture content both in offline and online modes, and you will also get the complete access of study materials too, both for online and offline students as well. So whoever wants to know more about these programs or interested to join in these programs, you can call us to the given number, or you can visit our institute offline branch directly, which is located in Narayangada, Hyderabad. So yes, let's get started with biomolecules, that is nucleic acids. We have already dealt with carbohydrates, lipids and proteins. Now here we are discussing about the last biomacromolecule, that is nucleic acid. So as we all know, nucleic acids are well known as what beta? Nucleic acids are popularly known as what? Polymer of nucleotides or polynucleotide chain, fine. Usually nucleic acids, best example we will be learning as what? Either DNA or RNA, okay, DNA or RNA. These are the nucleic acids we will be learning more. DNA or RNA, what do we call it as beta? Polymer of nucleotides. Both of this we will be saying what? Polymer of nucleic acids. Nucleotides, sorry, polymer of nucleotide, or you can also say it as polynucleotide chain, okay. You can also call it as what? Polynucleotide chain, fine. So here you look into nucleic acids. The best examples of nucleic acids that will learn is what? DNA and RNA, deoxyribonucleic acid or ribose nucleic acid. They are said as what? Polymer of nucleotides or polynucleotide chain. Clear ma? Here you look into, together with polysaccharides and polypeptides, these comprise the true macromolecular fraction of any living tissue or cell, okay. Along with polysaccharides and proteins, nucleic acids are also what? True biomacromolecules that land on what? Acid insoluble pool. So they are what? True biomacromolecules. True biomacromolecules. Macromolecules that land on acid insoluble pool. Nucleotides are building blocks of nucleic acids, okay. Nucleotides are what? Building blocks of nucleic acids. That only we have learned here, right? Polymer of nucleotides. 3 chemical distinct components will be present in the nucleic acids, okay. 3 distinct chemical components are present. That is why we will call it as what? Heterocyclic compound. We will call it as what beta? Heterocyclic compound. And what are those 3 distinct molecules that we learn? Okay, any nucleic acid is a combination of what? Sugar, phosphate group and nitrogen base. It is a combination of sugar, that is pentose sugar, be it ribose or deoxyribose, deoxyribose. I will be saying you the clear differentiation in between these 2 sugars, and sugar, nitrogen base, phosphate group will be connected. Let us discuss on the linkages of this nucleotide, okay. So here what the gist of this is what beta? DNA or RNA is a polynucleotide chain or polymer of nucleotides. Nucleotide is a heterocyclic compound wherein it has 3 components. How those components are linked, I will be telling you now. So here first you look into the sugar, okay. Sugar can be what? Deoxyribose or ribose. Deoxyribose or ribose sugar, deoxyribose or ribose sugar. Ribose sugar will be ribose or deoxyribose, both of them are pentose sugars only. Both of them are what ma? Pentose sugars only, wherein it is having how many carbons? 5 carbons on total. So yes, you have here C1, C2, C3, C4 and C5. Total it is having 5 carbons. The main difference between ribose and deoxyribose is at second carbon you will be having what? OH group in the ribose sugar. Oxygen, free oxygen will be present in the ribose sugar. This will make the RNA unstable, because in RNA the sugar molecule is what? Ribose. That will make the RNA reactive and unstable. Where deoxyribose, deoxy means oxygen is removed from the second carbon in the deoxyribose sugar. So what will be the structure of deoxyribose? It will be same as that of ribose, but then at second carbon C2, what will be there? H group will be there, OH will not be there. So this is about the sugars beta, ribose and deoxyribose. Now once the ribose sugar is connected to a nitrogen base, okay, if in nitrogen base you have to know what are the types of nitrogen bases first, then you can clearly understand about linkages. Now you have understood about sugar na beta, let's get into what? Nitrogen bases. As such we have how many? 5 types of nitrogen bases we have. How many? 5 types of nitrogen bases. Nitrogen bases. 5 types of nitrogen bases we have. What are they? 2 purines and 3 pyrimidines. Okay, purines and pyrimidines. Purines and pyrimidines. Purines will be having 9 nitrogens, okay, and it is what? Heterocyclic ring. It has 2 rings. It is what? Heterocyclic ring. Heterocyclic. And this is homocyclic, it just has 1 cycle and 6 nitrogens on total. So what are purines here beta? Adenine and guanine. Pyrimidines are what? Thymine, cytosine and uracil. Based on the amine groups and ketone groups or methyl groups present on these structures, their IUPAC names are derived. As such, adenine is also called as 6-aminopurine. Adenine is also called as what ma? 6-aminopurine. And guanine is said to be, so adenine here you can see na 6-aminopurine. Guanine is said to be what? 2-amino-6-oxypurine. 2-amino-6-oxypurine. What? Guanine. And as such, thymine is said to be, thymine is also called as what? 5-methyl-2,4-dioxypyrimidine. Okay, so here you can see 5-methyl-2,4-dioxymethylene. Okay, 5-pyrimidine. 5-methyl-2,4-dioxypyrimidine. Clear ma? So here it is. Thymine is what? 5-methyl-2,4-dioxypyrimidine. Cytosine, if you see, 4-methyl-2-oxypyrimidine. Okay, 4-methyl-2-oxypyrimidine. So here you can see cytosine is what? 4-methyl-2-oxypyrimidine. Next, if you see uracil, it is 2,4-dioxypyrimidine. Okay, uracil it is 2,4-dioxypyrimidine. Fine, and cytosine also, this is clear. Right, this is all about adenine, guanine, thymine and uracil. Most importantly ma, with the structures you learn its IUPAC names. IUPAC names are very important. Clear? Now you have got clear idea between sugar and nitrogen base. So this sugar and nitrogen base will be linked to form what? Nucleoside. So how nucleoside is formed? Nucleoside is formed by linking what? Sugar and nitrogen base. Here you look into it beta, sugar and nitrogen base. So sugar here it is what? At C1 carbon of the sugar is linked to N1 of pyrimidine and N9 of purine. Fine ma. So here C1 is linked with what? C1 carbon of the sugar is linked with N1 of pyrimidine, nitrogen 1 of pyrimidine, and C1 of the sugar, if it is linking with purine like adenine and guanine, it will be linked to what? N9 of purine. Clear? So what bond is linking this nitrogen base and sugar? What bond is it beta? Glycosidic bond. What is that bond? Glycosidic bond. Glycosidic bond. Fine. And next, if you have to see this nucleotide, nucleoside we are calling it as, right? What is a nucleoside? Sugar and nitrogen base. As such, you will be having nucleosides like adenine combining with sugar. You will call it as what, ma? Adenine along with sugar is called as what? Adenine along with, let us suppose, ribose sugar. We will call it as what? Adenosine. Adenosine. Guanine along with ribose sugar is called as what? Guanosine. Cytosine along with sugar, ribose sugar, is called as what? Cytidine. Fine. Thymine will not be there in RNA. So what will be there in RNA in the place of thymine? Uracil along with ribose sugar is called as what? Uridine. So as such, what do you have? Adenine along with ribose sugar, adenosine. Guanine along with ribose sugar, guanosine. Cytosine along with ribose, cytidine. Uracil along with ribose, uridine. If it is in the case of deoxyribose sugar, then what you will call it as beta? Deoxyadenine nucleoside, deoxyguanine nucleoside, deoxycytidine nucleoside, deoxythymidine nucleoside. So you have clearly got to know about the nucleoside, bonds included, and terms that we are using for it. Now let us look into the nucleotide. So here you see C5 of the sugar, C5 of the sugar is linked with phosphate ion by what? Ester bond. Is linked to phosphate by what, ma? Ester bond. Fine. So here in a single nucleotide, you have how many bonds? Glycosidic bonds and ester bond, phosphoester bond. So here let me clearly tell you in RNA and DNA. Okay, in RNA and DNA. Clearly take a note of it, beta. So in RNA, if you have to take, first nitrogen base along with sugar. What do you call it as what? If nitrogen base is adenine, guanine, cytosine, uracil. Adenine along with ribose. Same the case. Fine. You will call it as what, ma? Adenosine, right? Adenosine. Guanine along with ribose, guanosine. Cytosine along with ribose, cytidine. Uracil along with ribose, uridine. Clear? So this along with what? This is a nucleoside. What is a nucleotide? Nucleotide is nothing but nucleoside along with phosphate group. Okay, nucleoside along with phosphate group will give you what? Nucleotide. As such, what do you get here? Adenylic acid. Adenylic acid. So here you can take it down, beta. Nucleoside where at C5 carbon of sugar is bound to what? Phosphate group. By what bond? Phosphoester bond. Phosphoester bond. Clear? As such, here what do you get? Guanylic acid. And here what do you get? Cytidylic acid, and here uridylic acid. As such, you take down, ma. So this is all about what? RNA's nucleoside, its nucleotides. Same case if it is in the DNA. You have to put just deoxy in front of adenosine. That's it. Here if the sugar is what? Deoxyribose sugar. Then it is what? Deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymine. Deoxy what? Thymine. Thymidine. Fine. So here it will be again what? Deoxyadenylic acid, deoxyguanylic acid, deoxycytidyllic acid, deoxythymidic acid. So this will be the nucleotides. What are the bonds finally involved, ma? The bonds involved are 2 to form one nucleotide. That's C1 and N1 of pyrimidine, be it thymine, cytosine, uracil. And C1 of and N9 of purines, that is guanine and what? Adenine. How they are linked? By N-glycosidic bond. What is the second bond that is in between C5 carbon of the sugar and phosphate group? Okay, that is what bond? Phosphoester bond. So this forms out to be one nucleotide. Clear? So this is all about nucleotide beta. Now let us look into that polynucleotide chain. Okay, here in our NCERT the clear example is given about nucleoside and nucleotide, taking adenylic acid as an example. So here you can see at second carbon OH group is there, means what sugar it is? Ribose sugar definitely. So here you can get adenine, that is nitrogen base, and sugar at C1 carbon and at C9 here. Clear, right, ma? And phosphate group which is attached, so it is adenylic acid. Clear, right, ma? And the nitrogen bases, it's synonyms. Now let us look into the formation of a polynucleotide chain. How a polynucleotide chain is formed, how two nucleotides are linked. Let us look into that. First, the nucleotide which has third carbon OH group, and here fifth carbon of the preceding nucleotide will be linked by phosphate ion. Okay, so here one ester bond is also already there between fifth carbon and phosphate group in the formation of nucleotide. Here again OH group of the front sugar, that OH group at third carbon will be there both for RNA and DNA. So the third carbon OH group is linked with what? Phosphate group. Okay, phosphoric acid. The linkage is in between OH, alcohol, and an acid to form what? Ester bond. As such, here one more ester bond is formed. That is how two nucleotides will be linked, and this is how a polynucleotide chain will be formed. So in a nucleotide chain, to form a nucleotide chain, in between two nucleotides, okay, in between two nucleotides, here third carbon OH group is linked with what? Phosphate group. This phosphate group is also already connected with fifth carbon. Okay, this linkage is what, Nana? Phosphodiester bond. Phosphodiester bond. Clear, right? This is how a nucleotide chain is formed. Clear, right? Because of what bond? Phosphodiester bond. Clear. Next, you have to look into what? Watson and Crick model. As such, now we have got to know how a nucleotide is formed, how a polynucleotide chain is formed. Watson and Crick has proposed a model of double helical structure of DNA by looking or understanding the X-ray diffraction studies. So here you look into Watson and Crick's model of DNA. Watson and Crick's model of DNA. So here you look into Watson and Crick's model of DNA. What is present over here? Watson and Crick model, that double stranded DNA, here you can see the double stranded DNA, one strand from 5 prime to 3 prime, another strand from 3 prime to 5 prime. Here if you can see, at fifth carbon phosphate group will be there, that is free, and at third carbon OH group is there. As two nucleotides are connected through third carbon, fifth carbon, third and fifth. So at 1 and 5th, at 5 prime and 1 will be free, and 3 prime and OH group will be free. This will be there for each and every polynucleotide chain, but in a double stranded DNA, two chains will be running anti-parallel to each other. They run what? Anti-parallel to each other. Can you see it better? So both the chains in a double stranded DNA, both the chains are anti-parallel to each other. Anti-parallel to each other. And here in this double stranded DNA, two nitrogen bases, Purine is connected to pyrimidine, pyrimidine is connected to purine. How they are linked? For example, adenine of this chain is linked to thymine by 2 nitrogen bases. Here adenine is what beta? Purine, thymine is what? Pyrimidine. Same the case with guanine and cytosine. Guanine, which is a purine, is linked with what? Cytosine. These two are connected by what? Triple bond. These bonds are what? Hydrogen bonds. Hydrogen bonds. Fine. So, two nitrogen bases of 2 strands of DNA are connected by what bond? Hydrogen bonds. As such, as 2 nitrogen bases at the center, they are getting connected towards the periphery at the sides or at the peripheral side. What will be exposed? Sugar and phosphate group will be exposed. So, that is why in Watson and Crick model they said that sugar and phosphate groups are the backbone of this double stranded DNA. So, in the backbone what will be present beta? Backbone of DNA, backbone of DNA is sugar and phosphate groups, sugar and phosphate groups. Fine, backbone of the DNA is what? Sugar and phosphate groups. Now, this thing is clear, right? Anti-parallel to each other, and backbone is sugar and phosphate group, and at the center nitrogen bases are connected by what? Hydrogen bonds. Third is complementarity, and the next fourth point is what? Complementarity. So, 2 strands are complementary to each other. How they are complementary to each other? Purine is connected to pyrimidine, pyrimidine to purine. So, base ratio is constant and it is unique for each and every species. Okay. So, here if you have to look into adenine connected to thymine, guanine is connected to cytosine. They are complementary to each other. The complementarity is in between what? Purine and pyrimidine. So, he said that number of purines is equal to number of pyrimidines in a double stranded DNA. Okay. So, what is the most important point here? Number of purines is equal to number of pyrimidines. And of all the points which are proposed by Watson and Crick, the most important point, or you can say hallmarks of their proposition, is base pairing. What is the hallmark of Watson and Crick's model's proposition? Hallmark is what ma? Base pairing. Base pairing is hallmark of Watson and Crick model. Base pairing is the main hallmark. Okay. Base pairing is the main hallmark for Watson and Crick's model. Clear, right ma? This is all about Watson and Crick's model. Next. And here one more important point that I have to tell you is the distance between 2 base pairs and what is the pitch over here. So, in the double stranded DNA, the most standard form of the DNA is called as B form DNA. Where in B form DNA you have to know about major groups and minor groups. Okay. The major distance is, this is called as what? Major group, and this will be minor group. And one complete turn, here one complete turn is from here to here. Can you see? One complete turn is formed. This one complete turn length of DNA will be 3.4 nanometers or 34 angstroms. Okay. One complete turn length is what? 3.4 nanometers or what ma? 34 angstroms. And here in the B formed DNA, you will be having 10 base pairs on total. How many base pairs? 10 base pairs on total. And the distance between 2 base pairs will be what? 3.4 nanometers or 0.34 nanometers or 3.4 angstroms. Remember that, that is very important. The distance between 2 base pairs will be what? 3.4 angstroms or 0.34 nanometers. One complete turn, or this is called as one pitch of B form DNA, that is 34 angstroms, and it will be having how many? 10 base pairs. Now, after learning Watson and Crick model more clearly, now let us look into the 3 forms of DNA, that is A form DNA, B DNA and Z DNA. A DNA, if we have to look into the helix turn will be right handed. helix turn is what? Right-handed helix. And the number of base pairs per pitch, or you can say per turn, per pitch or per turn, number of base pairs will be here 11 base pairs. And major groove, if you have to see, the major groove is narrow and deep, and minor groove is what? Wide and shallow. And occurrence, where do you see A type of DNA? Mostly in the dehydrated or low water content, where it is highly concentrated, low water content, there you can see what? The A type of DNA. And it is less common and less stable as well. B type DNA is more commonly seen, more stable. This is seen in all the human cells, most common in all the cells, eukaryotic cells you can say. And the minor groove is narrow and deep here. Major groove is what? Major groove is wide and deep. And base pairs per turn, as we have discussed, will be 10 or 10.5 beta, and it is a right-handed helix. Okay, it is what? A right-handed helix. Z type DNA is what? Left-handed, and base pairs per turn will be 12 base pairs, and major groove is flat and absent, minor groove is narrow and deep. Occurrence, if you have to see, okay, let's be back to the class. That major groove is what? Flat and absent, minor groove will be narrow and deep. Occurrence, if you have to see, where do you see Z type of DNA? High salt concentrations with special sequences of GC, guanine, cytosine, nitrogen bases. Biological relevance, they may play major role in gene regulation, or the stability is again less stable. More stable is what? Most stable is what? B type of DNA. These are the three forms of DNA: aDNA, bDNA and zDNA. Next, Chargaff's rule. Okay, so in Chargaff's rule, if you have to see, during 1950, where did Chargaff propose this rule, taking what as reference? Chargaff has proposed these Chargaff's rules, which are only applicable to double-stranded DNA. Keep this in your mind. This rule is only applicable to what beta? Double-stranded DNA. Akshaya Shivani, good evening ma. My name is Prasanna. I am Prasanna Ma'am. Okay. So yes, next, Chargaff's rule. Just continue with the class beta. This is very important with respect to biomolecules, nucleic acids. Yeah. So Chargaff's rule, this is double-stranded DNA. It is applicable only to double-stranded DNA, and he has proposed these rules taking what as reference? Taking Watson and Crick's model as reference. Chargaff has proposed these rules. These are applicable only to double-stranded DNA. So where did he propose? In the year 1950, Erwin Chargaff concluded that in any DNA molecule it might be, the amount of purines and pyrimidines are equal, as how we have learnt, because one purine like adenine is connected to what? Thymine, and guanine to cytosine. As such, to make them equal, like he has given, adenine plus guanine will be equal to thymine plus cytosine in the double-stranded DNA. The amount of adenine is always equal to that thymine, and the amount of guanine is always equal to cytosine, right ma? Next, base ratio. This is very important, you see. Base ratio, A plus T by G plus C, adenine plus thymine by guanine plus cytosine. This is called as what? Base ratio. It varies from one species to other species. It will be constant to one particular species, and this base ratio will be varying with different species. For example, if you have to take the base ratio for a prokaryotic cell, the base ratio for a prokaryotic cell, base ratio for a prokaryotic cell will be less than 1, that is approximately 0.99, okay, it will be less than 1. If you have to consider the base ratio for eukaryotic cells, okay, base ratio for eukaryotic cells will be more than 1, like you can say, like for mammalian cell it will be around 1.2, Remember this is most important. And next, the deoxyribose sugar and phosphate components occur nearly in equal proportions, okay, because sugar phosphate backbones will be there, number of sugars will be equal to number of phosphates also, because in each and every nucleotide sugar phosphate will be there. So these are the Chargaff's rules and propositions. This is applicable for double stranded DNA. Okay. So types of RNA, function and proportion in total cellular RNA. First, as I told you, this Chargaff's rule is applicable only for double stranded DNA. Is it applicable for RNA also? Question to all. Akshaya, even you can answer. So yes, you tell me, ma, Chargaff's rule, is it applicable to RNA also? No. Chargaff rule will not be applicable to RNA because RNA is not double stranded, it is mostly single stranded. Few RNAs which are like in reoviruses, wherein you can see double stranded RNA sometimes, but most of the cases RNA is what? Single stranded. So Chargaff rule is not applicable to RNA. When it comes to eukaryotic cells, when it comes to what? Eukaryotic cells, you can see three types of RNAs. Let us look into those RNAs in detail, which is most important. Where are they synthesized actually? Okay. So if you have to consider this as a nucleus, and this is what? Nucleoplasm. Okay, nucleoplasm, and this is nucleolus. Okay, this is nucleolus, and this is what? Nucleoplasm. Nucleoplasm. Fine. So in a nucleolus, what is synthesized, ma? In nucleolus, rRNA is being synthesized. How rRNA is being synthesized? By polymerases. I will tell you polymerases also. Nucleolus will synthesize what? rRNA. In a nucleoplasm, mRNA and tRNA will be synthesized. mRNA and tRNA will be synthesized. mRNA, which is in the nucleolus, is being synthesized by a polymerase, rRNA in the nucleolus is synthesized by a polymerase called as RNA polymerase 1. Okay. mRNA in the nucleoplasm is synthesized by RNA polymerase 2. tRNA by RNA polymerase 3. Fine. Remember this is very important. Connecting mol bio and biomolecules, I am teaching this. Okay. Here you see what are the major functions of mRNA and what are the proportions of total cellular RNA. So here if you see messenger RNA, that is mRNA, which is being synthesized in the nucleoplasm by RNA polymerase 2. This carries the genetic code from DNA to ribosome. Yes or no? Messenger RNA will transmit the information from DNA to ribosome. This is actively participating in what? Transcription process. What is its occurrence in the cellular DNA? You have three types, right? How much percent of mRNA is present in total? It is just 5%. Okay. Which RNA is highly abundantly present in the cell? It is rRNA, that is ribosomal RNA, whose main function is structure and enzymatic role in ribosomes. Third is what? Transfer RNA, soluble RNA or adaptive RNA. All these are synonyms of what? tRNA. tRNA is having soluble RNA or adaptive RNA. This will transfer amino acids to ribosome during translation. It is just 10 to 15% of total cellular RNA. Remember, where is it synthesized? By what enzyme? Is it by Pol 1, Pol 2 or Pol 3? What is its functions and what is the proportion of this total cellular RNA? All these are important, and Chargaff's rule is not applicable to RNA because they are single stranded. Remember that. So here you can see same bonds, okay, like to form a single nucleotide. Here you can see this is what a single nucleotide. In between sugar C1 and N9 as it is guanine, glycosidic bond, C5 and phosphate group, that is ester bond, and in between two nucleotides 3' OH and 5' phosphate group. They are linked by what? Phosphodiester bond. Same linkage will be there. Clear right now? This is all about DNA, RNA and nucleic acids. Clear? Tomorrow let us exercise the last part of this biomolecules, that is enzymes. Thank you. So before ending the class, I would like to tell you one more time about NEET World's special programs, like it's 1 step to medicine, 2 steps to medicine and power step to medicine, where all those are included with 333 per month fee only. And to know more about this, as I have introduced in the beginning of the class, you can call on to the given number or you can directly visit our branch. Lastly, I would like to give you a grand platform for all the students who are going to study on their own, who are planning for their self-study for NEET 2026. NEET World is here with you with our Telegram channel. Like daily quizzes will be asked in our Telegram channel for botany, zoology, chemistry, physics and maths. Live class schedules will be updated in the Telegram channel. Free resources will be provided for study material and question test series also. One-on-one faculty interactions will be there. If you drop anything in the Telegram group, we will be answering your doubts personally. And then you can call on to the given number to know more about the institute courses or the groups that you need. And to log in into this Telegram group, you can directly scan this QR code or you can log in through our official website, that is www.neetworld.in. So NEET World will be your ultimate destination for NEET, JEE, EAP set and CUET entrances as well. Thank you for today. We'll meet you in the tomorrow's class with the most interesting topic again. Thank you.